Welcome everyone to our last series of webinars of using science as a Jewish professional. My name is Rabbi Jeff Middleman. I'm the founding director of Sinai and Synapses, and I'm thrilled to be here with the AAAS Dialogue on Science, Ethics, and Religion and Clal, the National Jewish Center for Learning and Leadership, a partnership to be able to help Jewish rabbis and cantors and educators learn about some of the most interesting and exciting and dynamic science and to be able to apply that in their own communities. And we also have to thank the John Templeton Foundation for their support. It's now my pleasure to introduce our guest speaker, Dr. Marnie Gelbart, who worked for the Personal Genetics Education Project at Harvard University. She is leading initiatives for advancing national awareness about the benefits as well as the ethical, legal, and social implications of knowing one's genome, which is becoming more and more prevalent in our society. She's the scientific advisor for P GED's curriculum and leads professional development trainings and classroom workshops for teachers and students. She received her BS in biology from Harvard College and her PhD in molecular and cellular biology from the Fred Hutchinson Cancer Research Center. And Dr. Gelbart, we are thrilled to be able to learn with and from you here this afternoon the questions of genetics and personal genetic enhancement and our ability to be able to increase who we are, enhance who we are, raises some incredible opportunities, some incredible ethical challenges, and I know you are at the forefront of a lot of these conversations. So we're thrilled to be able to learn from you here this afternoon, and I'm going to turn the floor over to you.
Great. Well, thank you, Jeff. Thank you to Sinai and Synapses, Clal, AAAS, and DOSER for inviting me to be here. It's really a fantastic opportunity. I think I'm equally excited to be here to learn from you all and getting your thoughts in the discussion. So I will do my best to keep my presentation on time so that we can have plenty of time for some discussion. I'd like to start out before I switch my screen here, just to share a personal story about how I went from being a researcher in genetics to my current position at P GED and having more of a role in education awareness. And that's when because when I was a postdoctoral researcher here at Harvard Medical School, and that was when I became pregnant with my older daughter. And I'm Ashkenazi, and I went to my doctor for one of my early appointments, and they offered me carrier testing to see if there were any variants, particularly several that are prevalent in Ashkenazi populations, to see if there was, if I had any variants that I might pass on to my daughter associated with some serious diseases. And at that moment, I said yes, and they drew 15 vials of blood. And for me, that was a profound moment, a vulnerable moment, and one where I thought to myself, I have a lot of questions, but I have those questions because I have a lot of knowledge from my training. And it became a personal mission of mine, and one that really led me to switch career paths and come to P GED to be involved in sharing knowledge, raising awareness, and being a part of a much broader question about these new technologies and how they're used. So it's very special for me personally to be here as well as professionally. Okay, so this is the website of my group, the Personal Genetics Education Project. For those of you who are calling in, our URL is pged.org. We are based in the Department of Genetics at Harvard Medical School, and really we were founded about 12 years ago out of a sense that the field of genetics was moving so quickly. I think to many in our field, we've really been surprised that it has gone even faster than anyone thought, and that these technologies can be potentially transformative. Some of my colleagues think transformative for medicine, but we also know they raise a number of ethical issues, as Jeff mentioned, and genetics has a terrible history of terrible history of eugenics, which our Jewish community knows very well. And that was fueled by a lot of misunderstanding about the science and also horrible judgment that some groups passed on to others. And so P GED came into existence out of a feeling that scientists need to do much better this time in speaking up, in making sure our communities are informed, and informed so that communities can be part of a much wider dialogue about the benefits of technologies, the implications, so that the scientific community can hear voices from all communities around in our nation and around the globe about how these technologies are used. So just briefly, and I don't know if many of you are on the phone, but I just wanted to share some of the resources that we make freely available on our website. So if you can't see this, you can go to our website and download a series of lesson plans that we developed for teachers that touch on a number of different topics. I'm showing here, I think we have 10 or so that are online today, and we're currently working on a series on genetics and identity. And what I'm gonna focus on broadly today is 3 sets of technologies that are driving so much of what P GED is doing and bringing about major implications for individuals, their families, future generations, so broad societal impacts. The technologies that I'm going to be focusing on are technologies that are making it possible to know much more about our own genetic makeup. And here I'm showing with the symbol of pedigrees that are commonly used by geneticists and genetic counselors to look at inheritance patterns within families, and that we are walking around as individuals and individuals with families with DNA that brings pieces, that we carry pieces of DNA from our ancestors, and genetics is just one piece of the puzzle. So our identity, our health is shaped by so much more than our genetics, but our genetics is increasingly coming into that conversation because we can have so much more access to our genetic information. The second set of technologies I'll talk about is how how genetics is increasing the amount of information we can know about future generations, and even parents able to have a say about the genetic makeup of the children that they want to have. And I'll close with some of the newest technologies that have caught a lot of attention for genome editing and making changes, very deliberate changes to our genomes. So here, what I'm showing is the technological leaps in genome sequencing. So in 1990, scientists started working on the Human Genome Project. It took 13 plus years to be able to, and cost over $3 billion to have an initial map of the human genome to decode the genetic makeup of a single individual. Now about 25 years later from when this all started, we can sequence a genome in 3 days, and there's at least one company that you can get your full genome sequence for $999. The goal ultimately for this to be really clinically as useful as possible is for sequencing to be feasible, sequencing a genome within hours. And we are certainly not there yet, but the goal has to be to continue to drop the cost till genome sequencing is free, so that issues of access so we can alleviate issues of access, so that there's equity in how these technologies are available to our communities. There are some companies that have developed DNA sequencers, like the one shown here, that can fit in the palm of your hand. These, if you can see, this particular sequencer called the MinION can just plug right into the USB port of a computer. And some scientists think that that one day this kind of sequencer is going to be available at your local drugstore, so that that this is really has the chance to democratize how people are able to access genetic information. So what can genetic information do for us? I'll highlight a few examples of this idea of precision medicine and how genetic information can inform diagnosis and treatment of disease, as well as preventative medicine. Here I'm showing CYP2D6, which is an enzyme in the liver that helps to metabolize codeine into morphine. And there are many known variants, 100 and counting, in the CYP2D6 enzyme that exist in human populations that affect how well different individuals are able to metabolize codeine into morphine. For some individuals, they're very poor metabolizers, maybe 5 to 10% of us. And these are individuals who might go to their doctor, be prescribed codeine, and actually get very little therapeutic effect of the medication. And for others, maybe a few percent of people, they're rapid metabolizers, that they actually metabolize codeine into morphine so quickly that it actually can be toxic, even life-threatening. And so for these individuals, they should never, ever be prescribed codeine. And so having access to that sort of information can be very informative for those individuals. More generally, genetics can, I think we're really at the early days of understanding how genetics can inform the kinds of medication that may work better for us or be dangerous. So just at the beginning, but one area where this is really in application quite a bit right now is in treatment of cancer, to have more targeted therapies that are less toxic. These aren't cures, but I can say from personal experience in my family that they can offer, for some people, a quality of life that might not be possible with more toxic treatments like chemotherapy. And so there's a lot of hope that this will continue to grow. Here I'm showing you a little boy in Wisconsin named Nicholas Volker. He is a child who, like many others right now, or like many others today, have an illness that really defies explanation and definitive diagnosis by their doctors. Nicholas had an inflammatory bowel condition unlike anything his doctors had ever seen. He underwent over 100 surgeries by the age of 4, and the only thing the doctors knew for sure was that if they didn't figure something out, that Nicholas was going to die. And so this was back in the late 2000s, I believe. Or no, maybe a little later than that. But they did something pretty unheard of at the time, which was to try and sequence Nick's genome to see if, or a portion of Nick's genome, to see if they could figure out what was going on. It is like searching for a needle in the haystack, but they were able to find a mutation to make a causative link to his illness, and it pointed the doctors towards a treatment, a path of treatment, and because of that, Nicholas is alive and well today. Still has some consequences of his illness, but he is. I think this was life-saving for Nick. Now, this is, a pretty rare story. Nick is the first person ever where genome sequencing pointed not only to a diagnosis but to a treatment. This is a rarity, but it's where people in the field of genetics hope they can improve. Then I wanted to turn to what genetic information might do for healthy individuals. And there are many ways that individuals can access their genetic information, both through their doctor's office, through a research project, through, in the Jewish community, there are certainly community-based genetic testing initiatives, and also consumer genetics, where companies are marketing genetic tests to consumers. there are a number of these companies and a lot of variation in what they test for. Some test for serious health conditions, like inherited predispositions for cancer, heart conditions, Alzheimer's predisposition, to ancestry, to nutrition, and the lighter side of genetics of how likely, what athletic ability, what kind of sport I should put my 7-year-old in. There's a lot of, I guess I should say these tests are different in what they do. Some sequence an entire genome or a portion of a genome. Some look just at specific places in the genome that are known to vary frequently among people. And these are called single nucleotide polymorphisms, or SNPs. And there's certainly different ways that people respond to finding out about their genetic information, and people respond, and depending what they find out may lead to drastically different kinds of behaviors. So there are certain variants in the BRCA1 and 2 mutations that are particularly prevalent in Ashkenazi populations. And for a woman who finds out that she carries one of these in her genome, she might consider undergoing a prophylactic mastectomy. And for a woman who finds out she doesn't carry one of these three variants, there's concern, what does someone take away from this information? So, as a woman who tests negative for these 3 variants is still has a risk of developing breast cancer in her lifetime. She just may not have this set of inherited predispositions. And so the FDA is thinking a lot about how to regulate the access to, regulate what kinds of consumer genetic tests are being sold. And so that's something I think we'll continue to see developing over the coming years. And there's some questions about how other kinds of tests, one for athletic ability, for example, should be regulated, because genetics is one piece of the puzzle. Our environment, our life experiences, our access, it plays a great role in who we are. And so who should be deciding, what kinds of information people can seek out, and how useful that information is, and how reliable and predictive that information is. There's great variability. So this is something that's developing right now. One of the concerns that some people have about accessing their genetic information is how it might be used against them. And so here I'm highlighting a piece of legislation that was signed into law in 2008 by then President Bush, the Genetic Information Non-Discrimination Act, or GINA. GINA is a piece of legislation that took Congress 14 years to pass, but protects against discrimination based on a person's genetic makeup or family history, and it protects against genetic discrimination by employers and in health insurance. So there are certainly areas that are not covered by GINA, and but it is a start for thinking about how people can make use of their genetic information while avoiding some potentially unwanted consequences of stigmatization or discrimination. And this is an area too, there are people who are very interested in having GINA expanded to cover long-term care insurance, disability insurance, and so forth. There's also been some movement that could strip away some of GINA's protections, and so this is ongoing. To turn now to what we can know about the genetic makeup of future generations. I mentioned before my personal story with carrier testing. There's 18 carrier tests currently for for diseases or for variants that are particularly prevalent in Ashkenazi populations. There are tests for variants associated with diseases in other populations. And this kind of carrier testing, preconception and premaritally and post-conception, has been a part of why diseases like tay-sachs have really decreased significantly in Jewish populations, and this kind of screening and awareness about this. There are ways that genetics can be used also for parents to have some sort of say in the children that they wish to have. Here I'm showing an embryo that was created by in vitro fertilization at the 8-cell stage. And if you can see on your screen, a single cell is being removed for genetic testing. And once genetic testing is done on embryos, then doctors can decide based on that information which embryos to implant in a woman. And this technology, which is called PreImplantation genetic diagnosis, can be used to avoid passing on serious genetic disease like tay-sachs, and has been applied in other ways that I'll talk about in a moment. Another way that many women are finding out more about the genetic makeup of the fetus that they're carrying is through something called non-invasive prenatal testing, or NIPT. And this is a way of learning about the genetic makeup of a fetus from small fragments of fetal DNA that are normally circulating within the bloodstream of a pregnant woman. This cell-free fetal DNA appears as early as 5 weeks of gestation, and after a woman delivers the baby, the cell-free DNA disappears almost immediately. So it's a way to non-invasively get some information about a fetus that a woman is carrying. It can be used to look for extra and missing copies of chromosomes, such as in Down syndrome. It can be used now to look at smaller regions of the genome, but with less accuracy. And doctors tell me that this is one of the fastest growing medical tests, most rapidly adopted, and millions of tests have now been performed worldwide. There's women may choose to seek out this information for a variety of reasons, or not seek it out. And for some women who, based on what they find, may consider abortion, I think doctors really want people to know that non-invasive prenatal testing, while much better than previous screening tests, is not diagnostic. And if a woman is considering termination, she is strongly encouraged to go for confirmatory testing by amniocentesis or chorionic villus sampling, CVS. So all of these reproductive technology get at the idea that genetics is now putting in our hands capacity for selection of who is born and who is not born. And there are many, and it's getting late, but there's many different opinions on how, what kinds of diseases, whether early childhood or those that onset later in life, whether we should select for sex, physical characteristics. There's many different thoughts on how, on what kinds of selection is or is not okay, where to draw the line, who decides, and this is certainly something that I imagine may come up later. Okay, so I will speed up a little bit and just to talk about genome editing, which is now taking us from a place where we can read a human genome to take the information that a person is carrying around, and actually using a number of technologies called genome editing to change that information. And this is technology that One in particular you may have heard of is called CRISPR. It is really talked about a lot because of its ease of use and low cost. It has really revolutionized the kinds of genetic research that is happening in institutions like Harvard Medical School. There are many applications. One is that's being looked at in pigs is to increase or to see if pigs might be a suitable source of organs to address global shortages for organs for donation to people who desperately need them. And the scientists are looking at pigs because their organs are comparable in size to human organs, and so that might be a source. Scientists are using CRISPR to see if they can eliminate dormant viruses within the pig genome that might reactivate in humans, or any other immune factors that might cause rejection. There's another way is CRISPR, and I'll just go through this quickly, just that CRISPR might be used to control vector-borne diseases such as malaria, dengue, Zika, Lyme disease, and something called gene drives, and I'm going to skip over that in the interest of time. But really, people are thinking about how CRISPR might be used in humans as well, both in tissues of the body to address disease, to make genetic changes and repair variants that are causing disease, and to do that in a fully formed human, where we're making changes in cells that won't be passed on to future generations, and scientists think that they can do this. We are beginning to see reports where this is being tested in humans, particularly in isolated blood cells, making modifications in those blood cells, putting them back into the body, and there are clinical trials underway, and even a couple that have been approved in August and October for treating certain kinds of cancer. There's many conditions that could be treated this way, inherited forms of blindness, muscle diseases, blood diseases, lung diseases, liver diseases, cancer. The potential for treating disease is something that has people very excited about where this could go. But these technologies are also raising a lot of questions because of the potential for germline gene therapy, genetic changes that could be passed on to future generations. And so I'm just wrapping up here to say that this is the subject of a lot of ethical debate amongst many stakeholders, scientists included, about whether making genetic modifications to future generations for the purposes of treating disease, for the purpose of enhancement, where do we draw the lines and who decides? And I'll just say that that a panel that was convened by the U.S. National Academy of Sciences gave a yellow light to human embryo editing recently, earlier this year. They said, we're not going to say no. The initial thought was, let's have a moratorium so that we can think about it. And now the recommendation is that we need to proceed really, really slowly here. There needs to be a lot of public input, long-term follow-up, oversight, but let's see what we can learn from research when there's no other reasonable alternatives. But this, and only when it's restricted to genes associated with a severe disease. So this is this is starting to move ahead very slowly and cautiously, but it is something that we're starting to see some publications about. So I'm just going to wrap up and say this is my P GED team. Most of us are based in Massachusetts. My colleague Dana Waring is up in Maine. And then here's our website. And I hope, Jeff, that you'll send out my email address and links to our information to everybody here.